Welcome to The Explainer. Today, we're diving into one of the absolute most fascinating microscopic stories in the entire natural world. We're going to unpack exactly how flowering plants reproduce. And trust me, it is a wild tale full of complex cellular machinery, perfect timing, and just ruthless biological efficiency. I mean, think about it for a second. If you're a stationary organism, literally rooted to the ground, how on earth do you go about mixing your genes to survive, adapt, and eventually conquer almost every terrestrial ecosystem on the planet? It's this massive evolutionary puzzle. Animals can just, walk, fly, or swim to find a mate. But plants? They had to engineer a totally different, incredibly ingenious way to spread across the globe. So to figure out how they pull this off, we're going to break this journey down into 5 chronological parts. Flower anatomy basics, the stamen and microsporogenesis, the pistil and megasporogenesis, the double fertilization mechanism, and finally, post-fertilization and fruit development. Let's jump right into Chapter 1, Flower Anatomy Basics, setting our reproductive stage. When you look at the big picture of a fully open flower, you can clearly see that the male and female organs are coexisting in this beautifully engineered shared space. Up at the top, you've got the male anther supported by a slender filament. And then right in the center, you have the female parts. There's the stigma, acting like a sticky pollen trap, which leads all the way down the long style into the ovary, which safely houses the ovules. It's an incredibly neat, self-contained system. All right, Chapter 2, the stamen and microsporogenesis, which is essentially the male journey. When we zoom way in on a cross-section of that anther, this is where the magic really happens for the male gametes. Inside these tiny pollen sacs, there's an innermost wall layer called the tapetum, and it immediately goes to work. Think of these tapetum cells as kind of like microscopic nursemaids. They have this really dense cytoplasm, and their whole job is to provide crucial, rich nourishment to the developing pollen grains inside. And inside those sacs is where microsporogenesis goes down. It's a step-by-step cellular division. Basically, compactly arranged sporogenous tissue cells step up to act as pollen mother cells. Through the process of meiosis, they divide to form microspore tetrads. Then, as the anther matures and naturally dehydrates, those tetrads separate and develop into thousands of individual mature pollen grains that are ready to be released into the world. Moving on to Chapter 3, the pistil and megasporogenesis. This is the female side of the journey. The female organ, known as the pistil, provides the entire physical pathway for reproduction. It starts way up top with the stigma. Now, this isn't just some random physical structure. It's a highly specialized, usually sticky landing pad that's designed specifically to catch traveling pollen grains. Right beneath it is the style, which is this elongated, slender stalk acting like a corridor. And down at the very bottom is the ovary, that bulged part at the base that protects the ovules, which are also called megasporangia. So megasporogenesis is the process of forming the female gametophyte. Deep inside the ovule, a single, large megaspore mother cell with a very prominent nucleus begins to divide. Through a series of complex cellular divisions, it eventually transforms into this incredibly intricate, multinucleated structure called the embryo sac. But getting to that final mature stage, well, that requires a pretty dramatic thinning of the herd. And notice the ruthless biological efficiency here. The mother cell goes through meiosis to produce 4 megaspores, right? But 3 of them just degenerate and die off. Only one functional survivor is left behind. That lone survivor then undergoes sequential mitotic divisions, meaning it replicates its nucleus without forming cell walls right away, until it magically organizes itself into a mature embryo sac. This sac has 8 nuclei perfectly distributed across 7 specialized cells, including the all-important egg cell. Which brings us to the climax of the story in Chapter 4, the double fertilization mechanism. Okay, get ready for a massive evolutionary plot twist. When a pollen grain lands on the stigma, it sprouts a tiny tube that grows all the way down the style, enters the ovule, and releases not 1 but 2 male gametes into the embryo sac. This triggers double fertilization. It's a phenomenon that is totally unique to flowering plants. It's incredibly rare in the broader natural world, and it requires these 2 distinct sperm cells to do 2 totally different jobs at the exact same time. So here's how those 2 jobs break down. The first male gamete fuses with the egg cell, a process we call syngamy, and bam, that creates the diploid zygote. That's what will eventually grow into the new plant. The second gamete bypasses the egg, moves to the center, and fuses with two polar nuclei in what's known as triple fusion. This creates a triploid cell that becomes the endosperm. The easiest way to think of the endosperm is like a packed lunch for the baby plant. It's a tissue absolutely bursting with reserve food to nourish the embryo as it develops.
And finally, Chapter 5, Post-Fertilization and Fruit Development. Let's look at the results.
Right after fertilization happens, that single-celled zygote kicks into overdrive. It rapidly divides and morphs. First, it grows into a round, globular embryo, then it transitions into this really distinct heart shape, before finally developing into a fully mature embryo, complete with cotyledons, which are the seed leaves, and a primary root axis. Now, as that embryo is growing, the surrounding real estate is transforming too. The ovules themselves mature into seeds. And it's pretty interesting because some seeds, like peas, are what we call non-albuminous. This means they completely gobble up all of their endosperm-packed lunch during development. But other seeds, like wheat, are albuminous, meaning they actually save a part of that endosperm to use much later during germination. Meanwhile, on the outside, the wall of the ovary hardens and develops into the fruit wall, which botanists call the pericarp. But here is a fun fact to share at your next dinner party. Not all fruits are created equal. True fruits, like mangoes, develop strictly from the plant's ovary, just as you'd expect. But sometimes external floral parts decide to crash the party. Take apples and strawberries, for example. A part of the flower called the thalamus actually contributes to the fleshy part of the fruit, creating what we officially call false fruits. So what does this all mean for us? We've zoomed in on microsporogenesis, we've seen the sheer ruthless efficiency of megasporogenesis, and we've unpacked the incredible packed lunch strategy of double fertilization. But considering just how delicate and complex this microscopic dance is, how has this specific mechanism allowed flowering plants to so completely dominate our world? It's something to really marvel at next time you bite into an apple or see a flower blooming on the sidewalk, because those tiny, invisible cellular mechanics quite literally shaped the green earth we live on today.